string. Now, the difference between the three is the cytoplasmic staining. There's a neutral stain, there's a basic stain, and an acidic stain. The neutral is clear, and there are far less granules. And so one is a neutrophil. It has a multi-lobe nucleus with a pink cytoplasm. If we skip 3 and 4 for a second and look at number 2, what kind of cell is that? It's okay just to go ahead and say it.
Monocyte.
All right, that's a monocyte. Now, why is it not a macrophage?
It has an irregular nucleus.
Okay, so morphologically, it looks like a monocyte. It's not. A macrophage would be much bigger. It would have many more cytoplasmic granules. It's also, because of where it's at, we know it's in the blood. And typically, you find monocytes in the blood and macrophages in tissue. Now, what about 3? That's one of our granulocytes.
Eosinophil?
Right, that's an eosinophil. It has these bright red granules, but still has this multi-lobe nucleus. Which leaves number 4 to be what?
Basophil.
Basophil. Right, it says it has the same multi-lobe nucleus, but it has these dark purple granules. Now, the one that students get hung up on the most is 5. And so, what is that? Is that a lymphocyte or is that a natural killer cell?
Lymphocyte, because you can't see the granules.
Right, there's very little cytoplasm. It's mostly just barely around the nucleus. There aren't any cytoplasmic granules that are there. A natural killer cell would be larger. It would have granules that you could see and would stain differently. Now remember, this was part of the problem for scientists in the early 19th century, figuring out what these cells did. Most people thought they were pretty unremarkable, most likely on their way to senescence. And so we didn't really know that lymphocytes did much until the late 1970s. Okay, so we've gone through sort of what would be the best properties of an immune system. And then we've talked about the players, and now we're going to talk about the playing field, or how the immune system circulates. And so it's really, this is more about physiology, about how the immune system is surveying for pathogens or infectious organisms. And the first thing we have to talk about are the different immune organs and how they're connected. So we differentiate these two different kinds of immune organs. There are the primary or central immune organs, and this is where hematopoiesis occurs and hematopoietic cell maturation. And so in an adult, that's primarily the bone marrow and thymus. In the developing fetus, this also can occur in the liver and some other sites. But for the purposes of this class, the 2 main central immune organs are the bone marrow and thymus. The bone marrow, of course, where the hematopoietic cell progenitors reside and where innate myeloid cells and B cells undergo maturation. And then you also have the thymus, which is a specialized structure for T cell development, which we'll cover quite a bit in this class. And then what you have are the secondary or peripheral immune organs. And this is where you initiate immune responses to infection. And so for the most important ones, of course, are your lymph nodes. And so if you were ever sick and your parent sort of felt your neck to see if you had a swollen gland, it's often called, they're actually looking for a swollen lymph node, which is indicative of an immune response to an infection. And you have many lymph nodes throughout your body that are all interconnected. You also have what is often called the lymph node of the circulatory system, which is the spleen. So this is where responses to bloodborne pathogens occurs. And then you have sort of more disorganized lymphoid tissues, which are the mucosal lymphoid tissues. And these are things like the Peyer's patches, tonsils, adenoids. These are associated with mucosal surfaces where we often get infected from. In many of the mucosal lymphoid surface or tissues, these don't actually exist until after infection. So it's sort of like a de novo lymphoid tissue. Now the circulatory system, where your blood is going through, is connected to the lymphatic system. And the cells, your immune cells, particular B cells and T cells, are constantly moving between the circulatory system and the lymphatic system. And we'll go over that more today. So let's start with hematopoiesis and primary lymphoid tissues. This is where you get the cells coming out. Now remember, your B cells and all of your myeloid cells develop in the bone marrow, okay? Now, we're gonna cover B cell development a lot more in the second part of this course, but what I want you to sort of focus on is where things happen right now. So, and we're gonna focus mainly on B cell progenitors, but remember, the myeloid cells are also coming out of the bone marrow. And in the bone marrow, this is where the B cell progenitors that have committed to the lymphoid pathway undergo rearrangement of their DNA to express a semi-random B cell receptor, so that each B cell coming out of the bone marrow has a different B cell receptor on it. Now, that's not sufficient, because if you can imagine, if you didn't have some selection criteria, some of those B cells would also recognize you and cause you to have autoimmunity. So in the bone marrow, those immature B cells that have rearranged their B cell receptor undergo a process called negative selection. And this is primarily so that you delete self-reactive cells. It doesn't get all of them, but it does get most of the ones that would recognize things that are in your circulatory system or that they would encounter in the blood. Now this is, because this is a central or primary lymphoid organ, this is often called central or primary tolerance. Central tolerance is the more common term. Now once those B cells mature and they've passed through negative selection, they exit the bone marrow by the central sinus vein. And so there's, I don't know if you've ever thought that your bones are connected to your blood supply. And that's very important for not only bringing oxygen into the bone marrow, but also for cells migrating out of the bone marrow to go and do their job. Now, the question really was, what do these cells do? Remember, they're small, unremarkable cells, and so it wasn't clear what their job was. And so early scientists used infection to test whether antibodies were being made, okay? And so they would take out an organ and say, are there any, is that animal susceptible to infection? If it's resistant, they were still making B cells, and so that wasn't the organ. But if they were susceptible to infection, that meant that they didn't have B cells, and therefore the B cells were coming from whatever organ they took out. This was really Bruce Glick and his trainees, Robert Good and Max Cooper. Max Cooper is still an active researcher at Emory University. And remember, in chickens, B cells came from the bursa of Fabricus, which is not the same as humans. In humans, it comes from bone, okay? And they could take out that bursa, just surgically remove it, and found that the chickens then became susceptible to salmonella, which meant the B cells were coming from the bursa, because the B cells are what confer resistance to salmonella infection. Now this was followed up a number of years later, or a couple years later, by Graham Mitchell and Jacques Miller. And they showed that if you irradiated the bone marrow in mammals, that you could destroy resistance to salmonella, and that if you put back bone marrow into those animals, you could restore that resistance. This is what it looked like. They would take mice and irradiate them. Irradiation, as you probably know, for cancer patients, will kill all rapidly dividing cells. It induces so much DNA damage that those cells die off. And that's true for the bone marrow. And that was known at the time, that you could use irradiation to destroy the bone marrow. And so if you did that, Oops. If you did that, then this mouse would have no bone marrow. If you injected it with salmonella, then it was susceptible to it and it would die. However, if you took the same irradiated mice and re-injected them with bone marrow to restore that compartment, they then were resistant to salmonella. And that meant that bone marrow in mammals was the site of B cell development. Okay, now remember that B cells are not the only cells to come from the bone marrow. Okay. Right, your myeloid cells, including your granulocytes, your monocytes, and dendritic cells and natural killer cells from the lymphoid lineage, they also mature in the bone marrow and enter circulation via this central sinus vein. The question is, do they undergo negative selection? Could you give me a thumbs up if you think they do? All right, I see a few thumbs up. Remember, how do these cells recognize things? Well, granulocytes and monocytes recognize things by conserved structures, right? So they're recognizing things that you don't have that are only present on foreign things. And so there's no need for them to undergo negative selection. Dendritic cells are sampling the environment, and so they also don't undergo negative selection. Their job is to present things, not to make a response to it. Now, natural killer cells might be the only one you would think might undergo negative selection, but natural killer cells recognize an imbalance of signals on an abnormal self cell. And they have just a couple of receptors to do this, so there's no need for them to undergo negative selection either. So all of these cells can just undergo development and then exit the bone marrow. So they don't have to do this central tolerance because they have very limited diversity receptors. And we'll actually talk about during T cell development that there's even some T cells that don't have to undergo negative selection. Okay, and so they're recognizing foreign structures. Those don't occur on your healthy cells in your body. Now, all of these cells exit the circulation into the tissues by this process of diapedesis. And we cover this more in later lectures. But diapedesis is the process of going from from the circulation into tissues. And it includes several steps, and there's sort of outlined there on the right. The first are low affinity interactions, sort of what's called tethering. And tethering is the process. It's just them sort of rolling along, sticking to stuff, and sort of moving on. And they don't undergo actually extravasation until they get a signal. And that signal is usually in the form of a chemokine. And so here you see their chemokine receptors binding to the chemokine, and that causes some other proteins to change conformation and to stick tightly to the cells. Now, following that, every time it gets a chemokine signal, it will increase binding in that direction and move towards the site of where the chemokine is coming from. Now, I don't care that you know all the steps in it right now. It's just that I want you to know that they go from the blood, they leave the bone marrow via the central sinus, they go into the blood, and then they move into tissues where they can do their job. And particularly for dendritic cells and your mast cells and then monocytes that are going to become macrophages, those exist in all tissues in your body because you have to be able to make a response there. Okay, so innate cells are present everywhere in the body. Neutrophils and granulocytes primarily are waiting in the blood and go to whenever there's a signal, but some of the cells just go there right after they develop. Now, T cells are also hematopoietic. And so the T cell progenitors develop in the bone marrow. And there's some signals that tell a lymphoid committed cell or lymphoid progenitor cell whether it should be a T cell or a B cell. If it gets a signal to be a T cell, it actually exits the bone marrow as a progenitor. It doesn't undergo any more steps and migrates to the thymus. Remember, the thymus sits right on top of your heart. It's a two-lobed or multi-lobed organ with a specialized job, which is just T cell development. And they do this by structures called high endothelial venules. And we'll talk a lot more about those sort of, as we say, talk about how cells move in. But these are specialized structures. They're highly invaginated, so they have lots of surface area, and they have a specific set of molecules or proteins on their surface that allow a T cell to know, yes, I need to go into that tissue. They also are expressing a chemokine that will activate the proteins that recognize the proteins on the high endothelial venules. Now, these are present in lymph nodes as well. And so there's a specialized structure designed for lymphocytes. Now, once the T cells get into the thymus, they then undergo somatic recombination of their DNA to express a semi-random T cell receptor. So that all the T cells coming out of the thymus have a different TCR on their surface. Now for T cells, remember T cells or T cell receptors recognize peptides that are presented by MHC proteins. And so you have two steps to their tolerance. First, you have to select everybody who actually can recognize an MHC protein, because you can imagine if it's a random DNA arrangement, many of them won't. So they don't pass that first step of being able to recognize an MHC with a peptide on it. The next step is to delete all of the cells that recognize MHC that have self-peptides on them. Okay, so there's a positive selection and a negative selection, whereas B cells only have a negative selection. Now, once those T cells pass those two steps, they exit the thymus and go into the blood, where they are going to start circulating, looking for their antigen. And that's what we'll spend most of the rest of the class. Now, it turns out the same groups were figuring out what T cells did. Now, but except the difference was that they didn't use susceptibility or resistance to salmonella, they used tissue rejection. And so if we go back for a second, MHC, or major histocompatibility complex proteins, were defined by, if you had the same MHC, you wouldn't reject an organ. But if you grafted on skin graft or tissues that had different MHC, the organ would be rejected. And so they use that tissue rejection to test whether T cells are being made. If T cells are there, animals will reject transplants from an unrelated donor. So from a different species or a different, not species, but a different breed of the same species. But if T cells were gone, then you wouldn't have anything to reject the graft, okay? And so the graft would be accepted. And so it's the same group. By this time, Bruce Glick had retired, so it was just Robert Good and Max Cooper. But they said, okay, if I take a chicken and I remove the bursa fabricus, the tissue still gets rejected. But if I take out the thymus, which is called a thymectomy, or thymectomized chickens, then the graft is not rejected, indicating that the T cells came from the thymus. If you remove the T cells, then the graft is not rejected. Well, Graham Mitchell and Jacques Miller saw this, and they did this essentially the same thing, but in mammals. And so they showed, again, that, that you could use bone marrow, you could radiate mice, destroy the bone marrow. That meant you got no T cells. But if you put bone marrow back, you got T cells. But if you took out the thymus when you restored the bone marrow, then you didn't get T cells. So there's sort of an extra step here, right? They would irradiate mice and put bone marrow back in to the mice. That would restore their hematopoiesis and certainly their B cells. If they didn't take out, if they didn't thymectomize them and did a skin graft, that graft will get rejected or killed. Sorry, there's an extra thing here. I'll have to take that up. But if they thymectomize the animals, then there's no place for the T cells to develop, and so the skin graft would not be rejected. Okay, so what I would encourage you, I'm going to ask you one of these two sets of experiments on the test. Tell me, how did they figure that out? How did they figure out that bone marrow was necessary, but also thymus for T cell development? Are there any questions on those two experiments? Okay, we'll keep going then. Okay, so that's basically all we're going to talk about for right now about central hematopoiesis and development of your B cells and T cells. Now the second question is, how do those cells that come out of the bone marrow, and we're going to limit it mostly to B cells and T cells, how do they go and look for antigen? How do they sense whether there's an infection? Immune responses always start in your secondary lymphoid tissues. So probably one of the main concepts that in particular physicians who haven't spent a lot of time in immunology forget, that you don't just make a response at the site of infection. It has to go to your secondary lymphoid tissues where you initiate that immune response. Then those cells, once they've proliferated and differentiated, will migrate out back to where the infection is and do their job. So that's where we talk about this delay in the adaptive immune response. That's where it comes from primarily. Now, the proof of this really was, or the question really was, well, what's happening? And there's two prevailing ideas at the time. One is that hematopoiesis was just pumping out cells. They would go out and eventually die. But then the other idea was that these cells were recirculating between the blood and then go into lymphoid tissues and then come back out into the blood. And at the time, they knew that the lymphocytes were going from lymphoid tissues into the blood via the thoracic duct. But what James Gowans did, he's a British researcher who had the idea to say, well, what if I put them back in? Does that restore them? And so what he did is he taught himself how to cannulate the thoracic duct. So the thoracic duct comes out of the lymphatic system, and it sits right on top of the heart. And if you cannulate that, you can actually, which is the process of sticking a rubber tube in there, you can collect everything that comes out of that duct and put it into a tube. And so he did that, and he said, well, look, if I take them all out, the number of white or lymphocytes in the blood goes down. But if I put them back into the blood via a vein, then the numbers stay constant, which means that they must be going from the blood into the lymphatic system, back into the blood, back into the lymphatic system. And they're constantly recirculating. Okay. Now, I'm sure most of you are familiar with the cardiovascular system. You have your heart, you have blood vessels, and here we typically draw arteries in red and veins in blue. We all know that's not the case. But what you may not know is that the lymphatic system mirrors your cardiovascular system. There's no heart, though. The lymphatic system is driven by peristalsis, or movement of your muscles that pushes fluid along the lymphatic system. Okay, so all of your hematopoietic derived cells initially enter the blood. This is true for cells coming out of the bone marrow, and it's true for T cells coming out of the thymus. And they recirculate between the blood and the lymphatic system. And we'll go into a little more detail how they do that. But essentially what's happening is that every capillary bed of your cardiovascular system also has capillaries of your lymphatic system. Okay, and so there's a system for collecting extracellular fluid and stuff that's getting sort of sloshed around at these capillary beds. you can think of it sort of like the drains, right? The arteries are pumping stuff in and the veins are taking stuff back. But it's quite a messy process, and a lot of fluid gets pumped out, right? Because in these capillary beds, there is some permeability. And so that fluid is then collected into lymphatics. And all of that fluid and everything else contained in that fluid is pumped by your movement towards the heart, so that it eventually ends up in your thoracic duct. Oops, sorry. Now, lymph nodes are the primary sites of infections to, or primary sites of immune responses to peripheral infections. And so if you look at where they start, basically from your knees to your elbows to the top of your head. You don't have lymph nodes in your hands or your feet because it's draining stuff from those peripheral sites. And so your popliteal lymph nodes, which are behind your knee, are responsible for everything that's downstream towards your foot. So everything that, if say you got infection in your pinky toe, that immune response is actually going to happen in a lymph node that's hidden behind your knee. Okay, so the idea here is that your lymphocytes, and primarily, are going to exit the blood, they're going to go out to peripheral tissue, and then they're going to enter into the lymphatic system. So lymphocytes don't typically go from arteries to veins, they go from arteries to lymphatics back to arteries. Whereas your red blood cells are going to go from arteries to veins. Now these drains that sit at every capillary bed are sort of look like very similar to your cardiovascular capillary, but they're intermeshed in between all of those so that they can collect everything. And the flow is towards a lymph node. Okay, so they're collecting stuff and your movement of your muscles is pushing that towards the lymph node. And so it really is this sort of three-way intermeshed capillary beds. And this is everywhere that you're pumping blood to, you're also draining from. Now the ends of these capillaries have lots of openings. They often call this a button junction. So if you think about a shirt with buttons, you can their stuff can move inside of the shirt by going between the buttons. And so it's an easy way to think of it. Now the drawing is not the best because it's actually not this little tube-like structure. It actually looks like a spread out maple leaf, like a real drain that you would see on the street sewers or something. Okay, so there's lots of surface area for stuff to move in. And entrance into these doesn't require any specific molecular interactions. And that's not entirely true for big things like cells, but certainly it's true for fluids and microbes, your proteins like chemokines and cytokines, soluble antigens. So anything smaller than a cell can freely enter it. Cells do have an extra step, but we're not going to cover that in this class. You can just think of these as drains for everything that's in that tissue. Now there's two terms that students often get confused on. It's sort of like, do you effect or affect, right? So afferent lymphatics are leading to a secondary lymphoid tissue. So if it's in your big toe and it goes to your popliteal lymph node behind your knee, that's coming into the lymphatic by the afferent lymphatics. But these lymph nodes are all connected. And so it can go from one lymph node to another lymph node. And when it leaves a lymph node, that's an efferent lymphatic. Okay, so it's the same thing as affect versus effect. Affect is something affects you. If you want to do something, then you have to effect it or effect it. Now, I have a Great Dane who's currently whining to go outside, but I like this picture because it's the best one to show how the lymphatics move. So the lymphatic flow is typically to the nearest lymph node. Now it can go from that lymph node to the next one, but that's called the draining lymph node. So we go back to the infection in the big toe example. The draining lymph node is the popliteal lymph node. That popliteal lymph node is connected to your inguinal lymph nodes, which is right at your hip. And then that's connected to, subsequently, to other lymph nodes and eventually goes to the heart. So there are a series of these lymph nodes set up all around your body to collect from certain areas of your body, okay? So your antigen and antigen presenting cells enter that draining lymph node from whatever region they started in. If the dendritic cell started in your thumb, then it's actually going to go into the lymph nodes in your elbow and eventually into the brachial lymph nodes in your armpit. Okay, and that's how you get antigen into the lymph node. Now the T cells and B cells don't come in that way. This is just how stuff from the periphery gets into lymph nodes. Now there's a few exceptions, right? We talked about the spleen. And so in the spleen, it doesn't have the same setup. The spleen, basically everything comes in through the splenic artery and everything leaves via the splenic vein. So it's a little bit different, but that's because the spleen's job is to survey the blood. And so there's no reason to have a separate system to pump the blood in and out of the spleen. The other one is mucosal pathogens, which have special transporter cells called M cells, in particular that sit over Peyer's patches, and they're just sampling the environment and they're directly pulling it into the Peyer's patches. So there's a little bit different setup in mucosal sites. The majority of this class, though, we're going to focus on lymph node responses. Now, as I said, the antigen and inflammatory cells or dendritic cells all get to the lymph node from the lymphatics. The lymphocytes don't enter the lymph nodes the same way. They go in through these high endothelial venules, which are these specialized structures that uniquely express a carbohydrate that allows cells to stick to them, okay? And so they come in through the high endothelial venules, but they can also go to once they do that, let's say it went to the popliteal, you don't want it to sit there forever. It spends about 24 hours, or for B cells, about 36 hours, and looks for antigen. If it doesn't see it, then it leaves the lymph node to go to the next lymph node. And so it's going out the efferent lymphatics and into the next lymph node by the afferent lymphatics. So if you remember, we said that lymphocytes don't spend most of the time in their blood. They're going from lymph node to lymph node to lymph node to lymph node. They don't see anything, eventually they're going to get dumped into the blood via the thoracic duct, and then just start the process all over again.
Is there a reason why we don't want a long residence time there?
Well, it's just a matter of surveillance. So, you know, it's a 1 in a million shot that you're going to have the right T cell for that antigen. And so you don't want it, you know, it's sort of like, well, if the police only stayed at the police station, they wouldn't be doing a very good job. So it's just they're circulating, they're spending some time in a lymph node looking to see if they see anything. If not, then they just go to the next one, they look there, they go to the next one, and then they look there. So let's talk about lymphatics for a minute. Many of you have probably taken a plane and you feel like after you do your plane ride, your feet are swollen, the shoes don't fit very well. Well, and the solution for that is for people to sort of do stretching exercises and flex your feet. What that's doing is increasing peristalsis and pushing the lymph out of that tissue upstream into the lymphatics. And this often can be seen in people who have impairment of their lymphatics, either through disease. There are certain infections that block lymphatics, and so you can see this elephantiasis, where you get swelling of the tissue. But you also see it in morbidly obese people. If you've ever watched My 600-Pound Life, right, and you think this person is just basically walking to the mailbox and they lost 60 pounds. And they often call that water weight. Well, it's not water. What it is they've basically pushed the lymph fluid, the extracellular fluid, back into the lymphatic system. And so and eventually that gets cleared out through the renal system. So it's really just pushing water or lymph back into the system. Well, what might be a main problem with that?
Wouldn't it overload their system if there's too much fluid?
Yeah.
That's a lot.
That's
Yeah.
exactly right. If they're pushing that much fluid, 50 pounds of fluid, that's a lot of fluid to put into your circulatory system, right? And so their blood pressure shoots through the roof, and suddenly they've got to take care of that problem. Okay, so it can be quite dangerous. Now, this can be due to lack of movement, right? If you're sitting on a plane and you're not moving your feet, fluid just accumulates there, and so your feet feel swollen. But it can also be tissue damage, like if you get into an accident which severs those lymphatics, there's no mechanism for you to drain that peripheral tissue. And then there's some infections that will intentionally block it. There are some people that have a defect that makes them not very efficient in lymphatic drainage. And so those people typically have what's called epidermal hydrolol, I forget. Basically, it means water skin, where you get little blisters that are essentially micro accumulations of lymph. So a lot of what we know, and I'm going to sort of take a sidetrack here, is that you can actually, a lot of what we know about lymphatics now and how they operate is you can inject dye into a peripheral lymph node and see where things move. What this is mostly used for is for people who have cancers, right? So a cancer in a peripheral organ can metastasize. Well, the way that it does that primarily is by entering into the lymphatic system, just like all of your other cells, and then moving around the body. It gets pushed by peristalsis and eventually goes into the blood. And so if you have a cancer, you inject this dye into the cancer, and then you look, does that dye end up, is it staining a lymph node blue? And so you can do this. This is sort of, you find the cancer, or in this case, the lymph channel, and then that leads upstream to this blue lymph node. And it's a way of detecting where, if the cancer was going to go somewhere, where did it go? So here's our picture, right? We have our circulatory system that's pumping the blood out through the arteries, through the capillary beds, and then coming back through the veins. The dendritic cells, your antigen-presenting cells, some of the inflammatory cells that are going to activate the immune system are going to the lymph nodes via the lymphatic capillaries, okay? Now they enter the lymph node by the afferent lymphatics, and then they can go out through the efferent lymphatics, which then eventually become afferent lymphatics for the next lymph node, and so on. But eventually, they're going to dump into the thoracic duct. And this is one of those cases where you are not symmetrical. Your 75% of your body is served by the, 75% of your body drains into the thoracic duct, but 25%, your upper right quadrant, drains into a very similar duct called the right lymphatic duct. In either case, all of the cells are going back into the arteries, okay? Now the T cells and B cells don't come into the lymph node typically directly via the lymphatics. They come in through the high endothelial venule. So if you were to track a T cell response, a T cell is going to go from the blood into the lymph node via the high endothelial venule. Now, its return is via the lymphatics. And so it can move through the lymphatics once it gets there, but it makes this kind of trip. Now, that's different than your red blood cells. Your red blood cells are going around here and coming around through the veins. Okay? So you have sort of three different patterns. Cells from peripheral capillary beds go directly into the lymphatics. T cells and B cells enter the lymphatics by the high endothelial venules. And then your red blood cells and platelets are circulating through the blood constantly. Now, once you actually make a response, there's something else happens here. And we will cover this quite a bit more when we actually talk about T cell responses to infection in the third part of this course. But this process of T cells leaving, going upstream to the next lymph node, well, it takes quite a long time. And that would slow your immune response down. So if you do make a response, if that 1 in a million T cell sees its antigen in the lymph node, it's actually going to make a response, and then it's going to exit by the high endothelial venules, because that's a direct line to getting back in the blood. You don't want it to go back through many more lymph nodes, because that would just take forever for the T cells and the B cells to get out and do their job. Okay, now I think we have to talk now about high endothelial venules. So just like any other part of your body, your lymph nodes require oxygen, nutrients, and that's carried by the blood. The high endothelial venules, seen here, are these little beam-like structures that are post-capillary beds. So here's our artery coming in. It changes to a vein because there's a capillary bed right here. And there's a structure that is on the post-capillary side of the venule that is highly invaginated. And so if we draw it, it's sort of like, it's got lots of invaginations, so there's lots of surface area. I know that's a pretty terrible job. But think of a mitochondria, how that looks. This is the same structure, but in a multicellular structure in your veins. Okay, and these HEV, or high endothelial venules, are specialized structures. They uniquely express a carbohydrate called sialyl X, which is only expressed in high endothelial venules and allows lymphocytes to stick there. And that's where the B cells and T cells will go from the capillary, or from the high endothelial venule, into the actual lymph node structure. Okay, so that's a high endothelial venule. And in this case, there's 3 of them. And it's basically, you have one for each trabecula, or each part of the lymph node. Now, stuff coming from, let me take off this pin. Stuff coming from the tissues, that are macrophages, dendritic cells, granulocytes, things that are going to activate the immune system. This is from an older review article, and it actually says lymphocytes. That's not how lymphocytes get to lymphoid organs. Even if they're memory cells, they're still going through the circulation. On the other hand, your lymphocytes are going to enter the lymph node by the high endothelial venule. Now, in reality, this shows granulocytes and other things. It's really these guys, the lymphocytes. These cells don't, which tells you, I think this review is from 2011. It's only 9 years ago, and we've changed a lot of our ideas about how the immune system works. Okay, so your activated lymphocytes stay in the blood circulation, or they're exiting via the blood, because that's where they want, if you made a response, you want to get them into the back of the blood as quickly as possible. Whereas the other lymphocytes that did not get activated, they leave the lymph node via the efferent lymphatics, because they're going to go to the next lymph node.
If these activated lymphocytes stay in blood circulation, are they using dipoiesis to then go to the site of the infection?
Yes, they are. But we'll talk about it. Your B cells don't. They go back to the bone marrow. Your T cells will go to the site of infection and start trying to kill it or do their job there. Other cells, T cells that get activated will stay in the lymph node, and we'll cover all of that in the third part of the semester. Right now, I just want you to sort of get an idea of how things are moving through the body. All right, any other questions before we move on? All right, so let's talk about the initiation of an adaptive immune response. And remember, this always has to occur in the lymph node or spleen. It's always the same rules, and I think people often get confused and think there's something different going on. So your immune cells are meeting in the lymph nodes, okay? And the main cells that initiate immune responses are dendritic cells. And for B cells, this is called the follicular dendritic cells. For T cells, it's called many of the other types of dendritic cells. Okay, so the first thing, here we have a picture of the infection in the lung, and you have dendritic cells that are in the lung. And so once they sense there's an infection, they will, by their PAMP receptors, that tells them, okay, you've sensed something foreign, stop penocytosis, because you've probably already picked it up, and migrate to the lymph node and try and present whatever you picked up to your T cells. Now they can also sense signals from other innate cells, for your granulocytes, your neutrophils that are exploding, or your macrophages that are making cytokines. That's the initiation of dendritic cell activation. Okay, so they stop penocytosis, because you don't want to present other stuff that you accumulate on the way to the lymph node. They upregulate your major histocompatibility complex proteins, so that they'll have lots of stuff on their surface to present to T cells. They also increase proteins we haven't talked about, which are called co-stimulatory proteins that are necessary for T cells to get full activation. And then they move to the lymph nodes. Your T cells are coming into the same lymph node by a different route, right? They're coming in by the high endothelial venules. Now, if they ran into the dendritic cell, but they recognize that's their antigen, they get all the signals that they need to get from the dendritic cells, they will then, inside the lymph node, start proliferating and differentiating into effector cells. Now, a lot of this is controlled by things that we'll talk about later in the semester about what kinds of signals they get. But the main outcome is you have activated T cells that are going to leave the lymph node now, go into the blood, and migrate by diapedesis into the tissues. If they don't recognize it, then they spend about 24 hours looking, they don't see their right antigen, so they just leave via the afferent lymphatics, or sorry, they leave via the efferent lymphatics to go to the next lymph node. Now, that's for T cells, but what about B cells? Okay, so for T cells, it's a little easier to think the dendritic cells just waiting there to pick up stuff and goes to the lymph node. For B cells, it's a little bit different. Now, typically, B cells are recognizing extracellular stuff, right? And so it can be soluble things, and we often call those particulate antigens, which just means that it's in the blood. And those can travel for the lymphatics, go freely to the lymph node via the lymph. But if it's anything bigger than a bacteria, it's typically transported there by macrophages, and they transport it on their cell surface. They don't engulf it. And so these macrophages have all of these scavenger receptors and complement receptors and antibody FC receptors that allow them to transport microbes intact to secondary lymphoid tissues or your lymph nodes. Now these receptors have two jobs. One, they activate the macrophage, but then they also tell it to go to the lymph node. Now, if the macrophage becomes activated, it can produce cytokines that are necessary for the activation of dendritic cells, right, that we just saw. Now, your B cells enter your lymph nodes, again, via the high endothelial venules. And so what's happening is the macrophages are actually transferring their antigen to follicular dendritic cells. And follicular dendritic cells are like Christmas tree of the immune system. They just hold stuff on their branches for long periods of time, and B cells come and sample it and see if they can recognize it. But it turns out this process, which was only described a few years ago, is very regulated. And so there are macrophages just inside of the lymph node that control the flow of stuff. And these are called subcapsular space macrophages. And they transfer the antigen to follicular dendritic cells, but they do it through this sort of messenger cell, which are just sort of lymph node resident B cells. These are not the B cells that recognize antigen. They're just recognizing stuff bound to the antigen and transport it. So they're just like the mail runners for the lymph node. Now, an important concept here that we'll talk about a lot this semester is that, remember, you can get a B cell response without a T cell response. It's not very strong, it's pretty weak. So a full activation of B cells and T cells requires both antigen on dendritic cells to activate T cells and antigen on the follicular dendritic cells to activate the B cells. And after that happens, this funny thing happens where the B cells go and talk to the T cell and say, hey, did you get activated? And if they did, then you get a full response. Now, if the B cells don't see anything on the follicular dendritic cells, they also go out via the efferent lymphatics, right? They go to the next lymph node and look around there and see if they can find it. Now, lymph nodes are highly organized structures, and they're organized into basically 3 zones. You have the cortical region, paracortical region, and the medullary sinus. The lymph nodes are organized into follicles, sometimes called trabecula, which are basically just sectioning out that lymph node. It's got one high endothelial venule for that part, and then everything is regulated within that sort of lobe of it. B cells typically occur in the cortical region. Sort of in this drawing, you can see B cells happen in the yellow part here. The T cells occupy typically the paracortical area, so this blue area. So B cells and T cells get activated in 2 different places. If B cells get activated, they form, what is called a germinal center. Okay, so in this case, this would be several responses to an infection, and there's several germinal centers. That's only present during the infection when the B cells are responding. Once the infection is controlled, then it senesces and it goes down in size and eventually disappears. Okay, so if there's a germinal center there, it's making a response to something. So remember, the T cells are coming in, they go in through the artery, through the high endothelial venule, which is in the paracortical region. And then this is the vein that comes out here. So the T cells go into the T cell zone as soon as they leave the high endothelial venule. The B cells have an additional trip to make. They actually have to go up here. So they're going to go to the B cell area. So B cell zones are in the cortical regions, and then they can form, they do recognize antigen, they get all their T cell signals, which we'll talk about again later in the semester. They proliferate and form this germinal center. The germinal center is where these B cells will start remutating their B cell receptor, selecting the best ones, and then making the right types of antibodies that are appropriate for that kind of response. Now the T cell zones are these blue areas. They're in the paracortical region, which, if you're up with your word roots, just means next to the cortical region. And these activated T cells will go to different places depending on what their job is. If they are helper cells, and particularly cells that we now know are called follicular helper cells, they will actually go to the germinal center and help the B cells. And that's a key part of a B cell response. If they're other types of cells, they'll actually leave the lymph node and go to where there's an infection or where they need to be to do their job. For CD8 T cells, remember they're killers, but they're much simpler. They have one job, and that's go to kill stuff. And so activated CD8 T cells just exit and go look for the infection. Now, if they didn't recognize their antigen, they were unlucky, they didn't see the right thing, they just go out via the efferent lymphatics, and ultimately this will dump them back into the blood so they can do this whole trip again. But if they did, the effector T cells and B cells don't want to take the time to make that long trip. They want to get into the blood and do their job as fast as they can. So they exit via the high endothelial venules. That puts them right back into the venous circulation.
So wait, just to be clear here, so since the T cells are activated by the conventional dendritic cells and the B cells are activated by the follicular dendritic cells, you should not expect to see, let's say, follicular dendritic cells in the paracortal regions.
That's correct. The FDCs will occur in the cortical B cell regions. Now, they're not static. They're moving throughout that region, and we'll talk about that later, but basically they're traveling all around there trying to maximize the chance that they're going to run into the right B cell. Okay, so there was a movie back several years ago where one of the superheroes was called the Spleen, and his superpower was to fart. I don't know what that has to do with the spleen. The spleen has a very specific job, or it has two specific functions. In the spleen, you have red pulp and white pulp. In the red pulp, that's where your red blood cells are tested for how old are they, do they need to be replaced, and if they need to be replaced, that's where they die, and you recycle stuff. And so the red pulp is a filter for your blood. It removes main job is to remove dead or dying erythrocytes. If you think about it, right, an erythrocyte develops, it dissolves its nucleus, and so it can't really make more protein, so it has a shelf life. And once that shelf life is over, it has to be replaced, and the way that you figure that out, just send it through the spleen. But the spleen also has this white pulp, and the white pulp is essentially has the same structure as a lymph node, but it's testing stuff that comes in through the blood. And so that's so that you can make an immune response to things that are circulating in your blood. And your spleen is fairly large. In humans, it's sort of like not quite football, but maybe a small Nerf ball size. And everything enters the spleen via the splenic artery. It's just a high pressure system. It sprays stuff into the spleen. Once it enters the spleen, it's going to enter into the red pulp. And then the lymphocytes will go from the red pulp into the white pulp, which is actually concentric around the artery. So in this case, your dendritic cells and macrophages are carrying antigen to the spleen, are going to enter the same way that the lymphocytes do. Everything comes in through the splenic artery. There's not a lymphatic system to deliver stuff to the spleen. But that's okay, because you're serving the blood, so makes sense for you just to look at the spleen or to have the blood dumped directly into the spleen. Now in the white pulp, it's organized into T cells and B cell zones. Looks just like a lymph node, but they're more about concentric sheaths around the splenic arterioles rather than trabecula. So they're sort of organized like a tube with a tube around it. Now, if the T cells and B cells go into the spleen, and they go through into the white pulp and they look for stuff, eventually they're just going to exit via the splenic vein. And so it gets collected by the splenic vein, which just basically dumps everything back into circulation. But if it gets activated, if you make effector T cells and B cells, how will it get out? Well, it gets out the same way. It just goes into the splenic vein. So everything is going in the same way and everything's coming out the same way. So if we update our diagram, we have this sort of other migration, right? Where a T cell is going to go out from the thoracic duct into the arteries. One of those arteries can lead to the spleen. It looks around here for a while. If it doesn't see anything, then it's going to come out the vein and then go back into circulation that way. Okay, so there's a number of different migrations, and please take the time to sort of diagram how it's moving here. Where are the cells coming from? Because it's important for the rest of the semester when we talk about making responses. Now, we talked about mucosal lymphoid tissues as sort of the third secondary lymphoid tissue, or peripheral lymphoid tissue. And these are more complex. And we don't spend a lot of time on this in this class. We do go into this in the advanced immunology class in spring. But essentially, they're organized very similarly, particularly the lymphoid tissues that are constantly there, which are the Peyer's patches, which are these little, they actually look like, they're named after bike tire patches, where if you look on the small intestine, there's little sort of raised circles on it that you can see with the eye. And Peyer just find these, and they were called Peyer's patches. They're organized into B cell and T cell areas. So if you look in a B cell area or in a Peyer's patch, it looks like it's flipped upside down, but it's essentially T cells areas are there in red, and then you have B cell areas in blue. Now, usually what's happening for all of those of you who had eggs this morning, your M cells are taking antigen into those Peyer's patches, and you're making a response to it. And we can talk a little bit more about why that is. But this is a way of you keeping all the commensal bacteria in your gut away from the epithelial layers. And they do this by making antibodies to the stuff that they're bringing in. However, that's usually a tolerizing response, meaning that it's not accompanied by inflammation, and so you're making antibody responses without really making a T cell response that's going to be, cause a lot of inflammation and damage. Okay, so now you've got your T cells and B cells. They're activated in the lymph node. And then they leave the lymph node by the high endothelial venule, and they go back into the blood and eventually are recirculating and looking for where they need to go to do their job. Well, first thing is B cells, the activated B cells that become plasma cells, move back to the bone marrow, where they spend the rest of their life just making antibody. For those of you who've done protein chemistry, they make about 1 milligram of antibody per cell per day. Now, that may not seem like a lot, but that's a ton of protein. And so they basically stop every other cell function, dedicate their entire rest of their life to just making antibody. The antibody is released and put into your blood, and so it can do its job. Now, effector T cells, cells that their job is to go and kill cells or go and help out with the response, they go to wherever the infection is, or into inflamed tissues. And this is controlled by those chemokines that are telling them, hey, there's something here, you should probably come here to do your job. But there's some other cells, right? If we can remember, part of immune protection is memory B cells. It's not just cells making antibodies. You also get memory B cells whose job it is to respond faster and better the next time you get that same infection. So how would they circulate? Well, they're going to circulate just like a naive cell so that they can go from lymph node to lymph node looking for, is there something to respond to? So they do that. They also have a second job, which is a little controversial. I'm not quite clear if these are a distinct subpopulation. Memory B cells will also replenish plasma cells that die, so that if your plasma cells die off, you're not going to lose immunity. They just get replaced by a memory B cell. We really don't know what the signal is that tells a memory B cell, yeah, you need to go and become a plasma cell long after an infection. But we do know that they can do that. Well, what about memory T cells, right? Most of the cells have died off, but you retain these memory cells and they continue to circulate in the blood just like naive cells. They don't replace, there's not a plasma cell equivalent for memory T cells to replace. Their job is just to go out and look if there's an infection again. If there is, then they can respond faster and better the next time you get to the same infection and make the response that much faster. Okay, so memory B and T cells are going back through lymph nodes. So we're going to end with bonus question number 4. When I was a graduate student, I had accidentally got stuck in the thigh with a syringe containing a live virus that I was working with. So the question is, if I stuck myself in the thigh, which lymph node would you expect to dramatically increase in size, given this sort of the lymph nodes that I have outlined here? And the second part of the question is, 10 years later, assuming I survived, which I did, would you expect to find memory T cells for this virus in my spleen? I don't answer it now, but this will be up on Canvas within the hour. And that's where I'm going to end and take questions if you have any.
I guess I had a quick question on the previous slide.
Sure.
When you initiate effector T cells, you recognize antigen, you're proliferating, is there like a population of those effector T cells that says, okay, we're going to become memory cells and then start circulating through the blood? Or is it the effector T cells, they do their function, they clear the pathogen, and then a few of them convert into memory? Like, when is the conversion of into memory? When does that happen?
So there is a point, and I'm glad you asked this, because this hasn't been a clear question, or it hasn't been a clear answer in immunology. I would say that up until about 10 years ago, or maybe 7 years ago, you would get immunologists who would say one or the other of those two scenarios. There's actually a couple other possibilities, but Sukek, when she was a postdoc with Rafi Ahmed, showed that memory precursors diverge during this proliferative phase, and they can be identified by some unique surface proteins, and they're not memory cells at that point, but they are memory cell precursors, and they sort of save themselves to become memory cells, and they don't become fully differentiated effector cells. Now, having said that, for B cells, that something similar happens, and during the germinal center formation, it's thought that memory B cells are actually exiting the germinal center a little bit earlier than plasma cells. And so for both of the memory B cells and memory T cells, it's during that proliferation that they're making a decision to become an effector cell or a memory cell.
Does that ever pose an issue if you make a memory cell and you're still actively fighting a function, and then the memory cell travels to another lymph node and recognizes a dendritic cell and then causes a second response?
Yeah, it's not necessarily a second response, but in terms of how you put it, yes, it's in chronic infections. What happens is if you start trying to make memory cells, you keep stimulating them and they keep differentiating, and eventually you don't have any more memory cells. And so what happens is that the response burns out. And this was what I did my PhD work on, and it goes through several steps, but it's called immune exhaustion, where your cells just keep responding and responding, and eventually they just can't respond anymore. And so what happens is some of them will die off, but many of them just go into this senescent state and eventually get cleared from the system, but it takes quite a long time. But it's very common in chronic infections. It happens in both hepatitis B and C. It happens in herpes virus infections. Debbie Van Barley showed that you, depending on how much stimulus you get, the more stimulus or the more chronic an infection is, the worse off you are. That's likely a big reason why it looks like COVID-19 infections are burning out, because that virus is actually can persist for 45 or 60 days. And so that's a long time for your immune response to keep trying to respond.
I also have a question about the circulation diagram.
Sure.
So it shows that high endothelial venules are entering lymph nodes and high endothelial venules are also exiting lymph nodes.
That's right. That's only if the T cells and B cells got activated.
Okay, so then they would activated ones enter from lymph node to lymph node, and then they exit the endothelial venule, so it's a more direct path to the next lymph node.
Right. So if a T cell, let's take two different scenarios here. T cell comes into this lymph node, and it looks for antigen. If it doesn't see it, where's it going to go?
I don't know. It's going to try to get to the next lymph node somehow.
Right. It's going to go this way. If there's no activation. But that going through all those lymph nodes takes quite a long time. It takes about 24 hours for each lymph node. So if this T cell came in here and it got activated, where is it going to come out?
The endothelial venule.
Right, it's going to go out here. So it doesn't have to waste all this time with all of those upstream lymph nodes.
Okay. Also, I thought venule referred to vein, but it looks like the top one is coming from an artery. Why is that? Is there a reason
if we go back, it's just because my skills are not that great. If you go back to look at where these are, they're blue here. So it's post-capillary, which is right here, where it goes from red to blue. It's on the venous side.
Okay.
And so that's a much lower pressure system. If it was on the arterial side, the pressure would be higher and it would be harder for the cells to stop and exit. Since it's on the venous side, it's much lower pressure and the cells can get out into the lymph node there.
Okay, so in that diagram that you have where it looks like it's coming from an artery, it's actually coming from a capillary bed?
Yeah, it's post-capillary.
Okay.
And if I was better, I should make the tips of these arrows blue, I guess.
Thank you.
So any other questions?
Professor, I had a question about how the exam was going to be administered, like if we were going to be able to use our notes or if it was going to be a closed note exam.
I'm talking with Evelyn about that today.
Okay, cool. Thank you.
Will there be short answers on the exam as well?
No, because of the format being online, we're going to have to do all multiple choice, true, false, matching kind of questions.
Okay.
I have sort of a tangential question, if you don't mind answering that. It's about the impaired lymphatic drainage we were talking about in morbidly obese people. So I was curious, is that when diuretics are prescribed to address hypertension, is that sort of the mechanism that they're trying to address, that impaired lymphatic drainage? Okay.
And in fact, bodybuilders will use this too. Just before a competition, they'll go on diuretics to sort of drain all of their extracellular fluid, and they can become hypotensive. It's the same idea.
Okay, thank you.
Okay, so there was a question if you need to know lymph node anatomy. My lymph node anatomy is not great. I'm more interested in that you understand the concept. If there's a particular lymph node that you would have to name, I'll tell you in the question. There was a question about does the adaptive immune response initiate at secondary lymphoid organs or the entire immune response? The initiation in lymphoid organs is only the adaptive response. The innate response initiates wherever the infection is. The virus that was in the syringe is called lymphocytic choriomeningitis virus. And it is just as awful as it sounds. All right, if there's no other questions, I see there's a question about bone marrow transplants. I'm not sure if that's what the question was, but yes, you need the bone marrow to get the immune cells. If you don't have them, then it doesn't work. All right, we'll see you all on Thursday.